Did I upload the last discussion?
No, sir.
I did not, right? Today I will upload both, okay? So, Sandar, you are right.
So,
So,
excuse me,
yeah,
sir.
yeah. Go ahead, Akansha.
So this is also to inform you, like lecture 17 that you uploaded had a lot of disturbance actually. So a lot of parts were not clearly audible.
Yeah, I understand that, Akansha, but the problem is that I cannot record the lecture. Like there was a problem in
No,
my
sir, not for
laptop.
re-recording. I just wanted to.
हाँ बोलो.
So like, no, I'm not requesting you for re-recording, but just I wanted to bring this to your notice. Like there was a lot
Yeah.
of disturbance, I think,
So
from the laptop.
I am reasonably confident there are many computer wizards within this group. If someone would take the pain of recording the video and cancel the noise, that will be great. It is not too complicated. Like during a period of silence, like when nobody is talking, that background noise will still be there. So that has to be done is capture that as the signature and deduct it from the overall video file from the audio component. Then it can restore to a large degree. If anybody has any experience of video or audio editing, should be able to do it. I just do not have the time. Otherwise, I would have been able to do it. Is there anybody in the group who has some experience of editing video or audio files? बहुत सन्नाटा है. Try it. Try it. Like there are many free softwares available where you can actually correct the noise in a audio file, uh, get the noise signature and deduct it from the overall sound file. Okay.
So,
Yeah.
I was thinking ki, like if I mean this is the case that in eukaryotes there are one part of the protein which is which can be responsible for some other function also.
Mm-hmm.
So maybe in the evolutionary process, like suppose like there is a protein which is containing multiple subunits,
Mm-hmm.
and this is having this protein has some function which is in prokaryotes. There is something broad kind of function which is in prokaryotes as well as in eukaryotes. But
Mm-hmm.
later on in the evolution, the organism became more complex, and some a function is required which can be done a small part of that protein, right?
Mm-hmm.
So, like this is how this happened. Like then a small part of that protein started functioning. It started doing that function which was required by the new organism which evolved from the previous organism,
Mm-hmm.
and this is how it went on, and like this complex mechanism came.
Yeah. So, like, let us take the easiest example, that gap DH, glyceraldehyde 3-phosphorodehydrogenase. Okay. Now, under normal circumstances, uh, when there is a signal that glycolysis has to happen, then you upregulate all the genes of glycolysis and you execute it. So if they are coming out of the same polycystonic RNA, all the proteins will be produced almost in equimolar amount simultaneously, and the catalytic process will be on.
Yes, sir.
But now, when you want GAPDH to function as a transcription factor, you do not want all the other genes of glycolysis to be turned on, right?
Right. Yeah.
So that flexibility is not there in the operon organization, which is there in the organization that is available in the eukaryotic genome.
Yeah. Okay.
So that is the way I look at it.
Yes,
Again,
sir.
that does not mean that this is the reason or the only reason, but one way of looking at it is this. Is that okay?
Yes, sir.
Okay. Who else?
Hello, sir.
Yeah, Ruchira.
Sir, so I wanted to ask this whole enhancer and mediator complex after the it has bound like the RNA pol has bound and it has started elongation. This whole complex generally disassembles or like
Yeah. So what happens is that the initiation factors, the GTFs, they remain anchored at the promoter only. It appears
okay,
that they do not dislodge. Okay. And
okay,
as the RNA pol 2 starts to move, the mediator complex also eventually disengage.
okay,
Okay. But then RNA pol 2 gets associated with other factors, elongation factors and splicing factors, etc.
okay. I wanted to ask because sometimes the enhancer is like a downstream sequence which may itself be part of the gene. So I wanted
Yes.
to ask like if it gets transcribed, so it needs to be free for that.
Yeah. So first of all, madam, enhancer is always part of the gene. If you go back to the first lecture, I defined gene that every cis element that is responsible for production of the RNA or the protein, whatever is the final product, is all part of the gene.
Yeah.
So, but I do understand what you were trying to say, that if the enhancer is in the middle, will it create a roadblock for the RNA pol 2 that is moving through it?
Yes.
Because if it is in the middle, it will be on the exon, and exons are transcribed as well. So I would say that I have never thought about it.
Okay.
No. And Abid will again ask me about that how to target LoxP sites, and I still have not gotten the opportunity to read it. I will read it. Okay. I
Okay.
will definitely talk about it before this semester ends or before the next exam is scheduled. Okay.
Okay.
और बोलो.
Sir,
Yeah.
in EMSA, so can you explain how these probes are made? Like they are radioactive, right? Like how do we
Yeah.
generate them? Can you
Yeah.
explain that a little bit?
That how we specifically phosphorylate 1,5 prime end.
No, actually, just a second. So I'm not talking about the DNase assay. For EMSA assay, we'll be generating probes which will be equivalent to the enhancer sequence, right?
Oh, so that is fairly straightforward. Okay.
Yes.
So the biggest challenge that we have, and you should understand, let me restate the question for you again. That is, whatever little bit molecular biology you have learned and the questions that I asked and you answered successfully almost all the time, that if I give you a signaling molecule or a transcription factor, and I say that how would you figure out what are the genes that are, let me try to draw it out, okay, because this is something that is very close to my heart. Okay. Let us take a signal transduction molecule. It will be easier. It will cover both. Say A is a ligand. Which binds to a receptor. and then the receptor gets phosphorylated. Then there is a transcription factor which gets phosphorylated. Then that moves to the nucleus, and then transcription happens, right? And transcription happens of multiple genes on multiple chromosomes, etc. Now let us say that within an hour you get to see that genes P, Q, R, S, T are upregulated because you added the ligand to the cell, right? And you can use microarray or RNA-seq to get up to this point, right, Navneet?
Yes, sir. Yeah.
Now you flip the question. Say you know that P, Q, R, S, T are 5 genes that are expressed in the pancreas just when pancreas is developing.
Okay.
Now you want to, and these are random genes, okay? Some enzymes, some transcription factors, some cell adhesion molecules, whatever. Now,
Okay.
say you want to tissue engineer pancreas, okay?
Okay.
Which lab are you in, Navdeep?
I haven't been assigned advisor yet.
Oh, so you are an MTech student.
Yeah.
Okay. So now you imagine that you have this. Now you imagine that you want to make pancreas in vitro. Because,
Okay.
if you can make pancreas, you can possibly have a dig at diabetes, right?
Yeah.
So you cannot force express so many genes. But if you know which one transcription factor or one signaling molecule, if activated, you will get all of this. You will be closer to making pancreas in vitro, right?
Yeah.
Now, if you only know that these are 5 genes that are expressed in pancreas at the very beginning of development, you want to know, and the fact that they were not expressed earlier or they are not expressed in any other cell type, so you know that there must be some tissue specific regulation, right?
Yeah.
How would
Yeah.
you know what is the regulator? If you know the output, how can you get to the input? See, when you know the input, you can always get to the output fairly easily, right?
Yeah.
If you flip the question, that if you know the output, can you predict the input? That's where molecular biology has not been able to make a dent yet. So
Okay.
now, as I have discussed in the subsequent lecture, that one way is to try to characterize each of the enhancers. Okay. Now, you know that this is where P gene starts from. Okay, you know the transcription start site of P gene.
Okay.
And you just take a hunch that it will be there within the upstream 10 kilobases. It's a hunch. Like I would say that it is the case with majority of the genes. But it is definitely not written on stone, and you have already seen with the example of PAX6 that how many different kinds of googlies can be there.
Yeah.
But you say you have a hunch. So then what you can do is you take this 10 kb piece, and that you can, because now the genome sequence is known, so you can choose suitable restriction sites to cut the fragment. You use a tiny part of it as a southern hybridization probe and see that where the 10 kb fragment is migrating, cut it from the gel and clone it, right?
Yeah.
Anyone who is not following, please ask me. Now you have cloned it. So now you have cloned that 10 kb. Now you can do further restriction mapping to know that there are restriction sites here, 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 here. And you can do you take smaller fragments, typically 1 to 2 kb fragments initially, and then you incubate it with an extract from developing pancreatic cells. Because if this is an enhancer for this set of genes, then the trans element should be there in the developing pancreatic cells.
Yes. Yeah.
And then if all these fragments are radioactive, and say you are doing let us name them: fragment 1, fragment 2, fragment 3, fragment 4, fragment 5. Okay, so you take fragment 1 plus extract, fragment 2 plus extract, like the lanes will look like this in your gel. Just fragment 1 plus extract. Okay,
Okay.
fragment 2, then fragment 2 plus extract. And then you see that in any case is the band actually shifting. You see here the band is shifting. So you know that there is something in the extract that is binding to fragment 2. Okay,
Yeah.
it may or may not be important, but you know that there is something. For all you know, that there is something highly positively charged protein that binds to any DNA piece. Okay, that is very much possible.
Okay.
But you will get something. Then you narrow down the sequence, then you take that 2 kb of fragment 2 and split it up further into the 300, 400 base pair fragments and see exactly where it is binding.
Okay.
That's how you will do it. But the biggest challenge comes here, that what if the enhancer is not in here? Then you are, again back to finding the needle in the haystack. Like it can be 2 megabases upstream, 2 megabases downstream, in the middle of the intron. You have no clue. If you get lucky, you will get something. Did
Okay.
I make it clear?
Yes, sir. Like I just want to clarify one thing. So these radioactive probes, they are linear DNA, right?
Yes, linear double-stranded DNA. Yes.
Okay, so yeah, that's what I wanted to ask. So if it's linear DNA, I thought there are chances that there is non-specific interaction. So I guess I kind of got that point too. So it can be non-specific also, right?
Absolutely, absolutely. The final, if you want to ask me, like if my lab is doing such an experiment, I will not be convinced till this happens. Say you got fragment 1A3 as the fragment to which you have shown with all sorts of controls that this genuinely binds a transcription factor. Okay. And it is likely to be, or maybe multiple transcription factors, it is likely to be the enhancer.
Okay.
I will continue to call it likely till I do this. I will take zygote, inject the fragment followed by a reporter, make a beautiful drawing that I have. Make a transgenic mouse.
Okay.
Then see if I can see the expression of the reporter in the developing pancreas in at least one of the lines. Until and unless I see that, I will not conclude anything.
Okay. So that will be the validation technique, like the final validation technique.
Yes, because see, the problem is that if you are doing it in cell culture,
Yeah.
You can use cell culture as a suggestion. Okay, like there are multiple levels of suggestion here, right? This itself is a suggestion that you propose that it might be there in the top 10 kb. in the upstream 10 kb. That is a suggestion. You are
Yeah.
getting a shift in EMSA. That is making the suggestion little stronger. Then you show that if you take any other piece of DNA, like, okay, so let me ask you this. Now that you guys have learned enough, your literacy level has quite gone up. Let me ask you this. This question is for Umar. Umar, what experiment can you do to demonstrate that this shift is because of the specific sequence of F1 and not some non-specific interaction?
Sir, DNA's footprint.
Okay, when you propose an experiment, please try to visualize the result. The question is that how would you demonstrate that this shift, meaning some protein or proteins is binding to F1 and that's why you are getting a shift, right? How do you demonstrate that this shift is because of the sequence of F1 and like specific sequence of F1 and not some random non-specific thing? If you do a footprint, if it is a non-specific interaction, you will still get a footprint.
Yeah. It will get this.
Anybody else?
Sir, can I try?
Priyanshu bolo.
Sir,
Yeah, who
if
is this?
Sir Shubhajit.
Bolo.
Sir, if we know the which transcription factor and if we have the antibody against that, if we bound the antibody with that transcription factor, then we will get another supershift. The band correspond another
Very good.
supershift band.
So, if you know the transcription factor that is binding to it, you will get a supershift. But that still will not prove the F1 is because of the specific sequence of F1. Your experiment will prove that the shift was because it bound to transcription factor X.
Okay.
Do you understand what I'm saying?
Yes, sir. Yes, sir.
So if we add a protease which destroy the protein, and
Yeah.
then we can
No, sir.
again.
Sir, can I try once again?
Yeah.
So we have to now we have to do a competitive assay. We have to add the F1 sequence without the radio level probe, and we
Yes.
will add in that complex in that mixture, and we will not going to see the band in the supershift position, because the DNA which is not radio labeled, that DNA will bind to the transcription factor, and as the that DNA is not labeled, we are not going to see any band, supershifted band.
Yes. It is competition. Still haven't proven that the binding is because of F1 sequence. You only proved by this that binding is indeed to F1. You will do the same thing. This is called cold experimental, and you do a cold control. Control, thinking about control is the biggest part of becoming a scientist. Okay. As I said multiple times, that data is always right. It is interpretation of the data that can be erroneous. And interpretation of the data is guided by the control experiments. Okay, I'll tell you a very silly, sad joke. There was this scientist who was trying to figure out, like he made an observation that whenever he plays music, this cockroach dances. So he tried to understand that how the cockroach hears, okay, listens to the music. So he first cut its antenna, the cockroach was still dancing. He cut its wings, the cockroach was still dancing. Eventually, he cut its legs, and cockroach was not dancing any longer. So he concluded that cockroach dances the ears through its legs. Okay, so that's what wrong controls can lead you to. So the right control here would be you take any other stretch of DNA of the same length. If F1 is say 2 kb, you take any other DNA of the same length, cold, and if the cold competition that you proposed here will not be able to abolish in this case, then you know that it is a sequence specific binding because of the TF1, because of the F1 fragment sequence, only the binding was happening. If you take any other DNA sequence, it will not be happening.
Okay, sir. Yeah.
Who did not understand? सबको समझ में आ गया. Very good.
Explain this once again.
Yeah, yeah, sure. So, let us say this is a protein, okay? And this protein binds specifically to this. Now, this is radioactive, okay? We call it hot F1. Now, what Shubhajit said is that if you take cold F1 in much higher amount, then eventually this one would be competed out and this will be bound. And this one will be somewhere over here, right? Now, because this is not radioactive, so you will not be able to see the shift because you are seeing the shift because of radioactive DNA, right?
Yes,
So,
sir.
आवाज दो.
Yes, sir.
Yes, sir.
You will not be able to see the shift. Now imagine that you still have the binding with the same thing, but this time you are putting in cold E. coli DNA. E. coli DNA is still negatively charged, right? The DNA chemical property does not change, right? So E. coli DNA should not be able to compete it out because this has no affinity towards binding. But if it is just a positively charged basic protein that binds to anything negatively charged, then this will be able to compete it out, right? So you will see competition going away with E. coli DNA as well. Then you know that it is a non-specific binding. Could I make it clear?
Yes, sir.
Okay. So that's what you do. Then you know that, okay, binding to this particular sequence is specific. Then you take up the next job to identify the protein or proteins that are binding to it. That is fairly simple. You use the piece of DNA as an affinity matrix immobilize it on column, and then you will get all the proteins bound to it. All sorts of proteins will bind to it, and you can send the whole thing for separate them on a gel, cut out for the bands from the SDS page, send them out for mass spec, and you will find there are say 3 transcription factors: TF1, TF2, the TFY. You found that this. So now you know the sequence, you know the transcription factors. Now next you have to know whether this is indeed important for the expression of this gene in the developing pancreas. So you now have multiple options. One is that if you knock out this, then the expression may go away or expression may get lighter. If you knock out both of these, then you might expect abolition of it. From the genome, if you only delete the F1 part and you see that the expression is gone, you know that this is the right thing. Or you make a reporter construct and see that the expression is happening in the pancreas. So nowadays, because these experiments are now much more feasible than before, so the demand on the scientist is very high, that will show the same thing from 20 different angles. Okay, so that's how you approach it. Is it clear?
Sir, how to check that F1, sir, F1, F1, F3, checking that this transcription factor? Sir, please repeat that.
I didn't understand, Kuna. Another time, please.
Sir, you say we have find that there is a 3 transcription factors. Then
Yeah.
how will you check that, sir? In like mice knockout or?
So you know about affinity purification, right?
Yes, sir. Yes, sir.
So you can covalently couple the DNA with agarose.
Yes, sir.
That will now become an affinity matrix, right?
Yes, sir.
Now you take extract from developing pancreas, pour it on that column, and then the proteins that are binding to the DNA, they will remain bound to the column.
Yes,
everything else will be washed off, right?
Yes, sir.
Now you boil it in presence of SDS gel loading dye and load it on a piece of gel.
Yes, sir.
And you will be able to see the bands.
Yes, sir.
You cut them out and send it for mass spectrometry, mass spec, which will give you the sequence.
Yes, sir.
That's all.
Sir, after that, how will you check that is this our perfect?
Oh, so you can then design multiple different kinds of experiments. One is that you can knock out those transcription factors in a mouse.
Yes, sir.
If they're, listen to me carefully, if they're non-redundant, critically important transcription factors, then if you knock them out, then the expression of PQRST in developing pancreas will be lost, right?
Yes, sir. Yes, sir.
But often they will be redundant. So then you knock out 2 genes simultaneously, TF1 and TF2. Then you will see that, okay, real reduction is happening. That is one way. The other way is that instead of knocking out an exon, you just do a regular mouse knockout, but this time you introduce the neocassette where F1 sequence was there, right?
Yes, sir.
So now the enhancer is gone. So now the expression of PQRST in the pancreas will be gone. And the final thing is that you take the piece of F1, make a reporter construct out of it, and make a transgenic mouse and see if you get expression of reporter in the pancreas.
Okay, sir. Yes, sir.
See, these are fairly straightforward things. There is not much complication. It is just that what I am asking of you is to think rationally and logical. That's all. It is not that, say, now you have been taught all the experimental techniques, right? So
Yes, sir.
it is just that you have to understand which technique can and should be utilized where.
Sir, like I have some problem. Like, where do you like, use for mice or when you use cell culture techniques? That like I have some
See,
difficulty to choose. Where do you,
if you ask someone like me, I will say that cell culture is always a mere suggestion. I'll tell you why. The best case, see, like between cell culture and an actual animal, there are multiple differences. One is the way cells get nutrition. In cell
yes, sir.
culture, they are in monolayer. They get nutrition from the medium all over it and abundant. In actual tissue, cells are in very complex 3D geometry. They are not directly in contact with blood. They get the nutrition through some passive diffusion between the bloodstream and the lymphatic system. Uh, uh, just the contact with other cells, like the 3D culture and 2D culture, that makes a whole lot of a difference. So
Yes, sir.
all of these taken together, cell culture is always a very poor substitute for in vivo. But because doing experiments in vivo is very time consuming and expensive, so
Yes,
you
sir.
can use cell culture as a first approximation. And then once you have gotten that, say you started with 500 candidates and you have now narrowed it down to five, then you can go in vivo and see which one of these five is right. But if you want to start with 500 candidates and everything in vivo, then your lab will have to have a $1 billion budget.
Yes, sir.
Yes,
Thank you.
So I have one question. Can one enhancer regulate more than one gene spatially or temporally?
okay. So I should confess that I am not aware of such an example, but
Okay.
I will not rule it out. Okay, you asked a very, very important question. So you look up this term on the net called super enhancer.
Okay, sir. Super.
Now, there is a massive debate going on in the scientific community whether there is indeed something called super enhancer, or is it multiple different enhancer elements that are working together. Like just to give you an example, Shubhajit, where are you from right now?
Sir,
Where are
I'm from Kolkata.
Okay, so when the Durga idol will eventually will be taken out for immersion, uh,
Okay.
one person will not be able to lift it up, right? You will need
Yes.
multiple people,
Right.
and their combined effort is called what is additive.
Right.
ठीक है ना? Like everybody is capable of lifting 10 kg, and 10 people can together lift 100 kg because of the additive nature of the effort. Okay, but like if you think of each one of these individuals as an enhancer element, an enhancer element is what that binds certain transcription factors, right?
Yes.
Now you think that the DNA and the chromatin conformation is such that multiple of these enhancer elements can interact with each other. So now the mediator complex has multiple different interaction surfaces, which can increase the interaction between the RNA pol 2 in the promoter, because multiple different enhancer elements are positioning it in the right place. So that is one thing. It is those who are proponent of this, they are simply saying that there is no such thing called super enhancer. It is just additive effect of multiple enhancer elements. Some people are saying no, that is not the case. There are multiple different enhancers, and together they become one enhancer, which is a super enhancer that regulates multiple different genes. But I do not know what is the last word about it. It can go either way. The debate is very hot and only 3 to 4 years old. But if this is true, then I do not see any conflict of one enhancer element being recruited in additive interaction with multiple different partners of enhancer elements. Okay. The original concept that a dedicated enhancer for a dedicated gene, even if that is flouted, the overall concept of transcriptional regulation will still remain intact. Do you understand?
Yes, sir.
Anything else?
Sir, I had one more question. So,
Yes.
in Saccharomyces cerevisiae, so they don't have introns, right?
Very
But
good. Yes.
but you have mentioned that like the eukaryotes evolved out of the operon system so that they can have multiple signals which will express different kinds of protein. So if that's the case, how is the variation produced? Like there can't be alternate splicing, right? So how is same protein produced because of two different signals different?
Okay, so that's where okay. So I think your answer lies here. Yeah. See the same gene, right? PAX6. Even if you forget the transcriptional variation, if you consider these two elements, one element can drive it for expression in pancreas, the other element can drive it for expression in lens and cornea, right?
Yeah.
So now if the cell type that it is in is pancreas, then it will be expressed. If the cell type that it is in, that is a lens primordia, then also it will be expressed. Any other cell type, it will not be expressed, right?
Yeah.
Now let us consider, because yeast is a unicellular organism. Now
Yes.
let us imagine that when you are growing it in presence of urea, I am completely making it up.
Okay.
Then you need, and this gene is not PAX6 and it does not have so many introns. This gene is X. When you are growing it in presence of urea, this X gene has a special moonlighting ability to degrade urea. Then you do not need this gene to be expressed, and its normal function is to catalyze conversion of a substrate A to B. So whenever you are growing it in A, then this enhancer element is used. When you are growing it in urea, this enhancer element is used. That is possible, right?
Yeah.
So that's how.
Okay, so
This variation gives us even more diversity and flexibility.
okay, so the protein product itself is not very different in both the cases, but its
Yes.
function is different.
That is exactly the case with engrailed or with all the moonlighting functions that we know of so far. Protein product per se is not different. You should read up about moonlighting function. It is, like if you want to, you should read up. It's very exciting. It is a new area of research. Very, very exciting.
Okay, sure, sir. Thank you.
So, like, does it mean that in the exam? Sorry, sir.
You got the term, right?
Yes, I got it. Yeah.
Yeah.
So, in the example, like, in which a promoter is used to grow yeast cells in the presence of glucose. I mean, the promoter is suppose galactose specific, so it allow the protein to express only in the presence of galactose, and not in the presence of glucose. So it is the property of the promoter or the enhancer.
Enhancer.
Okay, so this is not the property of the promoter.
Absolutely not. Promoter has the same property every time to recruit the RNA pol to it. The decision whether it should be recruited or not lies with the enhancer and the transcription factors.
Yeah, okay. And so one small doubt is there, like, which may sound stupid, but I don't know. I can.
Nothing is stupid. Nothing is stupid.
Okay. So like in the regulation of lag gene, when the cap binds to the cap site, CMP that binds to the cap site, and I mean the promoter is loose. The promoter binding is loose. That's why when cap binds, it make it tight, and then transcription happen. But sir, when the after initiation, the promoter has to move, right?
Yeah.
The promoter has to move, but
RNA pol has to move.
Yes, sir. RNA pol has to move. Sorry, I'm not promoter. RNA pol has to move, and in that case, like the RNA pol will not be not remain bound with the cap.
Yes.
Cap region. So in that case, will it not be get loosed again? I mean, loosen again.
Okay. Like let us think about it this way: that cap protein, when it is not bound by cyclic AMP, it does not have the ability to interact with RNA pol.
Okay.
When it is bound by cyclic AMP, it has the ability to bind RNA pol.
Yes, sir.
I am making it up, okay? I am just trying to give you a logical explanation. I am not saying that it is factually correct because I have not exactly studied the detail of it, but I am telling you how it is most likely to work. Now, when you have very high cyclic AMP, meaning low glucose, then cyclic like cap will be first recruited to what is called the cap site, which is like almost like an enhancer, right?
Yes, sir.
And because it is bound there, now the local concentration of RNA pol to the promoter will go up. Now, whenever you have a weak interaction, if you increase the concentration, then the percentage of stably bound goes up, right?
Yes, sir.
Just because you are increasing the concentration. And here you are increasing the concentration by interaction with the cyclic AMP bound cap. So more RNA pol will be recruited to the promoter.
The interaction of CAMP to the cap site, that is increasing the concentration of RNA polymerase.
Okay, another time. CAMP, cyclic AMP, bound to cap.
Yeah.
Now cap is able to interact with RNA pol.
Yeah.
In fact, I would be even more categorical. I will explain this concept when we come to translation initiation in even more detail. Okay. So this if you think of this is a 3 partner interaction. One is cyclic AMP, other is cap, and the third is cap site. Right? When
yeah.
I can always argue that when cyclic AMP bound cap is recruited to the cap site, then it assumes a conformation which is conducive for its interaction with RNA pol. Is it not feasible?
Yeah, yes, okay.
Cap itself has a conformation.
Mm-hmm.
Cyclic AMP bound cap has a conformation which allows it to be recruited to cap site. Now DNA bound cyclic AMP loaded cap has a conformation that allows it to bind to RNA pol.
Yeah, yes, sir.
That will increase the local concentration of RNA pol near the promoter.
Yeah, okay. Yeah.
RNA pol will be recruited. Like nobody will argue that RNA pol before and after recruitment to the promoter will retain the same conformation. Now the conformation will change.
Yeah, yes, sir.
Maybe change in conformation will allow it to do two things. One, move with like a crawl along the DNA and disengage from the cap. That will give the opportunity for the next RNA pol to come and get recruited.
Yes, sir.
And the frequency at which the RNA pols are getting recruited to the promoter is actually the rate limiting step for transcriptional rate.
Okay, yeah, right. Like in the previous situation, when cap was not binding, the RNA polymerase come and it is not stable at the site. It comes bind and then lose again.
Exactly. Therefore, the probability of a productive interaction is low.
Yeah, yes, sir.
In presence of cap site bound cyclic AMP loaded cap protein, the probability of effective interactions go up.
Okay. And sir, is there any conformational change in the RNA polymerase which is allowing it to remain bound with the sequence?
As far as I understand, the moment it is recruited, it undergoes conformational change and it is ready to get disengaged from the promoter.
And does it remain the conformation change?
No.
I mean, during the
no. So in eukaryotes, we know it for a fact that, let me try to go there. Yeah. In eukaryotes, we know it for a fact that the RNA polymerase, as it is going from promoter to promoter clearance to facilitating capping, then to facilitating splicing, the phosphorylation status of the C-terminal domain, that is the largest subunit C-terminal domain, changes continuously. Now here, because it is phosphorylation, you can very easily capture that. In bacteria, you do not have that kind of ability to probe, but just because this happens in eukaryotes, you can guess that something like this might be happening in prokaryotes as well. Anna?
Yes, sir. Yes, sir. Yeah, got it.
Okay. Anything else?
In the slides, like the slide number 10, I couldn't understand properly.
क्या हुआ?
So, I mean, I can understand why in the high level of tryptophan, that RNA polymerase is not able to transcribe.
Okay, so if you look at bacterial transcription termination as a topic, forget about this. Okay,
Okay.
bacterial transcription termination requires a transcription termination signal. That signal is that the DNA sequence will be such that the RNA transcript will be able to form a hairpin loop followed by poly U.
Yeah, okay.
Now, when there is high level of tryptophan, then the leader sequence can acquire such a structure.
Yeah, okay.
But when there is low level of tryptophan, this structure is not acquired, something else is acquired. So therefore, the transcription is not stopped.
So how does it happen? Like when the high level of tryptophan is there, it will form that
When
hairpin
there
loop.
is high level of tryptophan, then the ribosome travels from here to here rather fast. Okay, so
Yeah, okay.
that
Okay.
does not allow enough time for the 2 and 3 to form a duplex.
Loop, yeah. Yeah, okay.
That's all.
And why in the high level of tryptophan
When there is
ribosome
when the tryptophan level is low, because there will be some pause, so 2 and 3 will have enough time to form a duplex, like a hairpin, like a duplex. Then this termination sequence is not being generated, so RNA pol will move through. That's all. So this is on top of the translation regulation, transcription regulation that is there. Remember, tryptophan operator, the operon is already a repressible operon to begin with.
Yeah, yes, sir.
So this is a second level.
Okay, yeah.
Like I would strongly suggest that you hear this particular lecture. I do not know who this person is, but very nice explanation he has offered.
Okay.
See, here he is saying, read in conjunction with row dependent transcription termination. उसका भी मैंने रेफरेंस दे दिया आपको, पढ़ो.
Yes, sir. Yes, sir.
Anything else?
Yeah, yes, sir. Okay.
आप सवाल नहीं करोगे तो मैं करूंगा. Ankit. Ankit.
Yeah, sir.
Explain.
Today I have connectivity problems, sir.
क्यों?
Sir, Wi-Fi is not working properly.
Okay. Is it working now?
Yeah, yes, sir.
You explain to me why SNFs were found to be part of the mediator complex.
Sucrose non-fermenting proteins. Sir, I was like last time also you asked this question like why SWI proteins were yeah. So I was thinking like SWI is actually the switch. Okay, so like it is found in the mediator so that it can switch between the like one state is when it is bound with the enhancer, one state is when it is not bound with the enhancer. So one is the active state, one is the inactive state. So I was thinking something on this line actually. Am I like going in the like
10 में से 3 दूंगा.
Okay.
अच्छा, आयुष बोलो, आयुष का भी आज नेट का प्रॉब्लम है, बार बार ही इज गेटिंग ऑफ एंड ऑन.
Yes, sir. I'm sorry for that.
बोलो.
So, I read about it on the internet and I couldn't find anything relevant. But as per the concept, since SWI-SNF is a chromatin remodeling complex, right?
Yeah.
Hello?
Huh? Go ahead.
Hello?
Yeah, go ahead. We can
Am I
hear you.
audible, sir?
Yeah.
Since SWI-SNF is an chromatin remodeling complex, as far as I know. So, what should be done is like in the chromatin in the DNA bound to nucleosomes or the histone proteins. What SWI-SNF might be doing is acting as a mediator. It allows the transcription to happen. I mean, I do not have a concrete answer for that, to be honest.
Okay. अंतरा बोलो. अंतरा को कभी कोई सवाल ही नहीं पूछा आज तक मैंने. Antara, where are you? In Calcutta?
No, sir. No, sir.
Where?
Sir, Chennai.
Huh?
Chennai.
In Chennai. Oh, then you are not cursing me. All the people who are in Calcutta, they are cursing me like crazy. But Kusat did not allow us to have fun outside. Bolo.
So, like, I'm not sure about the answer, sir.
Okay. Okay. Let me find the next person. Shreya. श्रेया किधर गया? भाग गई? Okay. प्रियांशु बोलो.
Yes, sir. So, what I think, because when a normal yeast cell utilizes glucose when the glucose is in the medium, but when it is provided with the sucrose, it needs an enzyme invertase to break it down into glucose and fructose, then it can utilize that. But since sucrose non-fermenting mutants do not have this function, so I mean the remodeling of this such that the transcription of this invertase gene can happen. Those factors are mutant or either absent. So from that, like we can associate it with the mediator complex, that these SNFs have a function in remodeling and also making those genes available for transcription.
आपको भी 4 दूंगा because both you and Ankit understood, but the clarity is still not there. I need more clarity. So Sandharv, you attempt the same thing with this question. Maybe it will give us all some clarity. With this thing, that why SRBs were part of the mediator complex.
So, like, I haven't reached that part of the lecture. I mean, haven't gone through those slides yet.
ठीक है, next quiz में questions सबसे tough दूंगा मैं. You guys are taking it easy.
No, sir, I like there were some net issues, so
I have barely slept. Okay. And I am still conducting my discussion hour when I am in Calcutta with my family, and sleep deprived, but I still consider it my responsibility to conduct this. If you are not putting in your hard work, I will raise the level of questions. Shubhajit, bolo.
So can you repeat the question once again?
What are SRBs?
Okay, sir. SRBs are suppressors of RNA pol B. So these are the proteins which are part of the mediator complex. And
how were they identified?
Okay, sir. They were identified. The RNA polymerase 2 has one subunit, the beta subunit, the RPB1, which has a C-terminal domain, has the heptade repeat sequence.
Heptade
Heptade
is
repeat
7. Heptade is 7. So 7 amino acid repeat sequence.
Repeat sequence. Yes, sir. The yeast cells which have mutation in that heptade repeat sequence. So those RNA polymerase 2 cannot involve in the transcription properly.
They cannot support activated transcription properly.
Yes, activated transcription properly due to there is some interaction with the CTD with the other mediator complex proteins.
Yeah.
So if there is any mutation in any of the protein of the mediator complex which are going to interact with the CTD, if that protein is going to mutate, then there will be no need of that interaction. So
or
then,
in other words, if you do not allow certain phosphorylation of CTD to happen, then it will not be able to interact with the normal mediator complex.
Yes.
But if some components of the mediator complex are by themselves mutated, then just like we have learned in the second lecture, that can induce conformational change in the mutant partner. Now they are again perfectly happy couple.
Yes, right.
So that's how you got the SRBs. Okay, suppressors of RNA pol B mutation.
Yes.
Why does it make sense for them to be part of the mediator complex? Because when they found the SRBs, they had no idea about mediator complex, right? You see, it is 1989 paper.
Yes, sir.
Okay, and the purification of mediator complex was if I am not mistaken, it will be 96, 97, something like that. Okay. So why did it immediately make sense to everybody that why it is there? Because the role of mediator complex is to support activated transcription. Right? When transcription is happening at a basal rate, you do not need the mediator complex.
No.
In each of these cases, whether for switch, normally the HO locus is not being transcribed, right? Only when there is a mating type change, then only HO locus has to be transcribed.
Yes.
So that function that will require participation of mediator complex.
Okay.
When you are transcribing invertase, normally it is not transcribed. Only when you have sucrose as the sole carbon source, then only it will be transcribed.
Yes.
So a normally quiescent promoter suddenly has to be activated.
Right.
There you will need the mediator complex. Therefore, a few of the mutants in each of these things mapped to the mediator complex. Is that clear now?
Not very clear.
Not very clear? Okay.
Yes, sir.
This is clear that the mediated complex is needed only in the activated transcription and not in the basal level of transcription.
Yeah.
Yeah.
So now, if you are looking for why some yeast cells are not being able to grow in presence of sucrose as the sole carbon source, then you think, what is it that will be needed? The gene has to be, the invertase gene has to be, like first of all, some sensing mechanism has to be there that the yeast is not in glucose but now in sucrose.
Yes.
So there will be some gene products required for that. Then the inverters, in response to that sensing, inverters will have to be transcribed.
Yeah.
Most of the cases, translation is not regulated for most of the cases. So translation we can eliminate, because any mutation in translation pathway, cells will be dead. Okay. Then there can be, like after translation, because invertase is a secreted protein, then it has to go through the Golgi, like ER uptake and Golgi.
Yeah,
and
the whole signal, the whole pathway. Yes.
So there will be some genes that are mutated. Like, I'm not saying there will be. If any gene involved in sensing the presence of sucrose is mutated, then also you will get an SNF phenotype.
Yes.
If any gene that is involved in secretion, not only for inverters, the secretion is a general pathway.
Yeah.
But if there is a defect in secretion in general, one of the output will be that it will not be able to utilize sucrose.
Sucrose, yeah.
Similarly, if there is an inefficient activated transcription, along with other genes, transcription of invertase on demand will also be affected.
Yes.
So that is the reason, and transcription of invertase on demand is dependent on presence of an active mediator complex, among other things.
Yes.
So if the mediator complex is not right, then you will not have them grow in presence of sucrose. For all you know, if you took those same mutants and acid for switch, mating type switch there, perhaps you would have seen that they are defective in mating type switching as well.
Yeah.
Every instance where activated transcription is required, the SNF mutants ideally should be compromised. Like these SNF mutants, like the specific ones that were mapped to like SNF 245. There might be SNF6 might be in the ER translocation machinery. SNF7 might be in the secretory machinery. But these 246 are in the 245 are in the activated transcription machinery in the mediator complex. So these three mutants, my guess would be that if you now challenge these mutants to undergo mating type switching, they might be inefficient there as well.
Okay.
So anything and everything that will require activated transcription, if any mutation is there in the mediator complex, then they will be inefficient in the process. That's all.
Okay, sir. Now it's clear.
Anyone else? Okay, so I am not happy. Okay, you guys are not studying enough. बंगाली वाला पूजा मेरा है और मस्ती आप लोग कर रहे हो. वो नहीं चलेगा. So you study and get into this part. Okay, we will discuss this part threadbare in the next class. In the next discussion. Our next discussion is when? 25th, na?
26th, sir. Monday, na? Monday, 9:30 a.m.
नहीं क्यों आज फ्राइडे है. अच्छा 26th Monday 9:30 a.m. right?
Yes, sir.
Good. So we will meet you on that day. ठीक है ना? Okay. But please study. हाँ, और जितने भी बंगाली थे सब भाग गया.
Sir, any idea when we will be having our next quiz?
Yeah. Oishi has the full calendar. Oishi, why don't you share that with everybody?
Yes, sir, I'll do that.
Yeah, share it.
Okay, sir. Thank you, sir.
Sure. Okay, chalo. Bye.
Okay, sir. Okay, sir.